Okay, hello everybody. And today we have a very interesting topic. Actually, we're going to talk about ATAC-seq, CUT&RUN, and promoters and enhancers. So let me share the screen. Okay, ATAC-seq, CUT&RUN, and enhancers. And my name is Alexander Kell, and I'm from geneXplain. And what we are talking about today is actually promoters and enhancers and how do they control gene expression. And this is a general schema. Of course, you understand this schema because you are here. You basically must be familiar with what is the transcription factors, how they bind to DNA. And so I'm not going to tell you these basics, just to remind you that what we call usually promoters are those regions in front of the gene, in front of a transcription start site, which are, harboring basic transcription factors and also other special transcription factors which regulate the activity of genes. And actually, RNA polymerase is sitting here and initiate the transcription. In addition, we have those enhancers and repressors, which are far away from promoters, and they cooperate with promoter areas, and they also harbor binding sites for transcription factors. And actually, in fact, there is still a question: where are the enhancers, and what is the difference between enhancers and promoters, and if there is any difference at all? So actually, if you believe there are some differences, so you see, I want to interact with you, so you see, I'm actually watching you, what you're doing. Let me just tell me in the chat, basically, do you believe that promoters and enhancers are the same things, or they are completely different? So if you think they are the same, just type 1. If they are different, type 2. And we will see and what are your answers, but I will just follow up. So again, if you think these are the same things, it's 1. If you think it's completely different, type 2. Okay, I see 1, 2. So 2 different meanings, and that's very good. So because in fact, I'm still not hiding. I'm hiding from answer because waiting for your guesses. Okay, let's see in the future. Maybe we can answer it later. So they're completely different. Okay, you think so? Again, different. Very nice. Let me see. Okay, so the consensus here that they're completely different. Okay, that's also a general belief. Although there is a little doubt behind that. But generally, there is let me just show you this slide from this paper, pretty old one, but this is a kind of a basic conclusions which were made in that paper. And people, and we are actually following this very consistently. Actually, promoters and enhancers, they are doing similar things. They actually contribute to gene regulation. So they contribute to regulation of switching on, enhancing the expression of the gene, or actually repressing expression of the gene. And but they have some special features. And if you look at the epigenomic features, which can discriminate promoters and enhancers, and meaning actually people looking at the special markers on the histones, whether it's methylation of special histones or acetylation, then depending what kind of marker you're looking, whether it's monomethylation or trimethylation or just monoacetylation, then you can see this difference in the signal. For instance, if you see that trimethylation of this histone of H3K27, then it's quite specific for promoter areas. But in case if they're repressed, actually this H3K27 acetylation is a bit more for enhancers. And it's the same, like if you see methylation, monomethylation of H3K4, It's also in enhancer and in promoters, but basically by combining these three, for instance, this one as well, so you can try to discriminate between them. So actually these markers can be used to discriminate promoters and enhancers, but actually in the end, they all are about binding transcription factors to their places. And then these transcription factors, by cooperating with other factors, they regulate transcription of the genes. And let's first talk about ATAC-seq. So, of course, I'm not going to introduce you this technology, just, from this very nice paper, for instance, where ATAC-seq was, reviewed. Basically, what you see is, in essence, you have a transcription factors, like here you see them like this little pictogram. They binding, for instance, in this place, in this case, in the promoter area in front of the gene. And then actually what they do, they use this transposase TN5, which cuts the areas of DNA which are opened. So which means basically for the transcription factors to bind, you have to have the chromatin open. So you have to have these nucleosomes be side a little bit, and this area should be free for binding of transcription factors. And then actually what you see then by putting this transposase, it will cut the areas of DNA which is free. And then when we have these fragments of DNA, we can sequence them, and then we can map them to the genome, and then voila. So those areas which supposed to be open, they will be sequenced. And so we see where the open areas of DNA. So that's like a first step. Before a transcription factor to be bind, the area should be opened from nucleosomes. But then actually also, which is shown here, sometimes you observe that those reads, which exactly covers the place where the transcription factor is binding, they can decrease in amount. You can have such kind of a valley here inside the peak, which may show you where exactly the transcription factors are binding. All right, so that's how this technology works. And today we will talk about pretty new technology, although it's quite well used already in many fields. And it's called CUT&RUN. And here, of course, people start using the very nice crisp system attached to various, types of design. And essentially what happens if you have a transcription factor sitting on DNA, then with a special antibody, you cannot avoid so far antibodies. Well, there are some attempts to avoid them, but you need an antibody which specifically recognizes the transcription factor, or maybe a specific, modified version of histone, if you look for histone markers. But if you look for particular places of binding for transcription factors, then we need an antibody for transcription factors. And then this antibody is attached to a special construct which carry these nice MNase, nucleases, which cut exactly the area where transcription factors are binding. And then we have these DNA fragments, which you can again sequence and map them back to genome, and you know where the transcription factor was binding. And essentially, this method is very good because it doesn't require this cross-linking of the transcription factor to DNA, which is usually done as a first step of a standard ChIP-seq experiment, which we haven't talked yet today, but you probably know about it. And this cross-linking process creates a lot of unspecific signal. And here there is no cross-linking. There's a clear cut. That's why it's called CUT&RUN. And that's why when you look at those signal, and what you see here is some area of genome. And if you look, if you do the ChIP-seq with this marker, then you get a signal like that. And if you do CUT&RUN, that your signal to noise ratio is much higher. So you see those peak. So basically the noise is much less. So which means You need less sequencing to get the same data type of data. And also the experiments gets much cheaper and cleaner. All right. So the next step actually, but we'll talk about that in a minute. So the peak calling. But we are not going to talk about peak calling today, about different type of algorithms. I just review it for you. I guess you might be interested in that. So there's plenty of peak calling algorithms. So once all these steps, after you have your sequence fragment and you map them to genome and you control the quality of this mapping, so you get this mapping on genome, and then you need to actually define the areas where DNA is, where transcription factors, for instance, is binding, or where the chromatin is open. So which means nucleosomes are not binding. Anyway, so in a sense, you need those peaks, which means the areas of interest. And then there is a number of peak calling algorithm, and I will review it in a moment. But what we are going to talk today is what happens after you have your peaks. Okay, so what? So you have your peaks, fine, but what to do next? And the next is actually the most interesting biological question. So if you have your peaks, then you may guess, especially if you do the peak, you do the experiment like ATAC-seq, you don't know in advance which transcription factor you are looking for. So you just have an open chromatin, right? And then you want to know perhaps which transcription factors are contributing to that regulation. And then there's several methods here. And one method which we will be describing today and discussing today is how to guess which transcription factor are binding to that region by enrichment of that so-called motif enrichment. So with that, we will use TRANSFAC to do the motif enrichment and to guess which transcription factors are there. And then by combining this transcription factor knowledge with RNA-seq data on another side, which tells you which genes are actually differentially expressed, we can really build the whole model of what's going on in the system. All right, just a little bit about the peak calling algorithm. Please don't bother too much in looking at this. It's again from that paper, in that review paper. There are a number of peak calling algorithms. You may see these familiar names like Max and Homer and other types of algorithms. They are based on different types of statistics. And actually, for the CUT&RUN, there is another interesting algorithm which is called Caesar, or ACR, whatever you can pronounce it. Yeah, but this algorithm is actually interesting because it's very simple, and when you apply it, it works for CUT&RUN better than, say, Max, although actually it has some it has some benefits, so it finds peaks better. But one problem is the peaks which it creates. If you analyze areas like, for instance, this methylation, trimethylation of the nucleosomes, then the size of the peaks is much bigger compared to other algorithms. So that you have to take into account, especially if you want to really focus your study on analyzing transcription factors. All right. Okay, so before we going now to the real example, let me ask you actually one question. We have prepared here pool, and I asked my assistant to put up this pool. So actually, I would like to understand what actually is your interest. So what kind of organisms, and whether you're working on just on human, or you're working also on mouse, rat, or maybe other types of model organisms. Maybe you're just working in plants. And everywhere in each of these organisms, you actually can apply ATAC-seq or CUT&RUN technologies. They all can be applied. And in all of those organisms, you actually can then apply TRANSFAC to find the enrichment of your motifs. So please answer this quiz. And when you're ready with your answer, you will see the results. So then in kind of let's do it in parallel. So please answer your those questions, and then you'll see the results. But anyway, so in our presentation today, I will focus on human. And the first example which I'm going to show is actually a very interesting data set where the study was done in acute myeloma leukemia. Actually, both of examples today, I will present two examples, and both of them will be on leukemia, on lymphoma, the second one. But in this example, it's a pretty new paper. Actually, these authors, they were studying this transcription factor, which is called BPTF. It's a pretty new transcription factor in a sense, new in terms of looking at it by researchers. It's involved in many cell signaling processes, and it's involved in we will look actually at this transcription factor more in detail. And what they did, they realized that actually this transcription factor is involved in developing of this acute myeloma leukemia. And actually, when they created a system, when they knock out this transcription factor, and then they see they're doing ATAC-seq experiment of the wild type of the cells of the acute myeloma leukemia cells, and after knockout of this transcription factor. And actually, after knockout, these cells are going to apoptosis. So basically, leukemia is, that's exactly what we want. So we want to kill leukemia cells. And that's happened when this transcription factor is knocked out. But we want to see whether those ATAC-seq area, they really contain these motifs for this transcription factor. All right, so for that, we'll go now to, let me stop the presentation. All right, so let's just go directly to our platform where we can put the data from this experiment. And actually we'll take, of course, you will see that these are huge data. So of course, these are a result of genome sequencing. So this size of the files are very big, like 3 GB and longer. So we will not do it like online, so like analyzing all these FASTQ files, but I will show how does it look like in the platform. So let's go now to the platform, geneXplain platform, where we're going to analyze this data. So for that, I hope you see my screen here. So we go to our website, and we will log into the website. So here with the login. So for the demonstration, I have my own login. If you don't have, you have to register here. So the access to the platform is free. So you can analyze your data, of course, with a limited size. If you have a license for TRANSFAC, you get a bigger space. You can also get this space on our server with additional. You can get additional space to analyze these big types of files. But of course, I have the space, so I can show you how to analyze those files. So now we log in, and here we have this geneXplain platform, and we go directly to the platform. All right, so so what you see here, if you are not yet familiar with this platform, you see here this nice 4 screens. And in the top kind of main screen, you see those types of data which platform can analyze. So, and today we are going to focus on well, it's called ChIP-seq, but basically here we have pipelines and methods to analyze all kind of epigenomic data, including ATAC-seq and CUT&RUN. You have here also for RNA-seq data, and even you now can analyze microarray data if you have on all kind of other types of omics data. But what we are going to do today is actually to look at this data here. So what we do, we actually go to this SRA archives. Simply use this run name, you know, and import this into the system. So they're importing by this button here, and then you have to put here in the row, you have to put this name of that on that run, and then it will be imported. And as you know, in the SRA system, actually, it's all recorded in a special compact format. Here, by importing, it's unpacking it, and we have special tools here to unpack it and to convert it to back to FASTQ files. And just additional kind of view on our platform. So you see here, the platform here has a number of tools for manipulation, for running this omics data. And we have also here the Galaxy tool. So the Galaxy fully integrated into the platform, geneXplain platform, where you can you do many Galaxy functions, including this you know like SRA toolkits, which allows you to convert SRA data into FASTQ data and other types of toolkits which Galaxy has. All right, so we converted them to FASTQ files, so they are here. And now, you know, as I told you in the beginning, what we need to do is to convert this into to map them first into basically genome. And for that, we have here in ChIP-seq, we have you know, once you load this, then we yeah, the mapping actually, I guess it's here in NGS. So you basically can align the FASTQ file using any of those tools. So you click on the tool, and then you drag and drop FASTQ file here. And here, since it's a paired end tools or append end sequencing, so you put both files here. So for the first run and for the second run. And then you define also the output, and it's a standard alignment algorithm, which then basically map those FASTQ files on the genome. And then what we got here, we got those BOM files here. And as you can see, we have here in the archive, we have 1, 2, 3, 4 files for the wild type and 4 files for the knockout. All right, so just remember, these are 4 and 5 and 10 and 11 for wild type, and 6 to 9 is for knockout. That's what we have here. That's why these BED files, which we have here, there are some coming from wild type, some from the knockout. And to visualize them, we have here in the platform a genome browser. And so actually you can see the genome with all the genes which are there. And here what we can do is basically just drag and drop any of those BOM files. So for instance, in wild type, so you see those reads here in the genome browser. So they cover the areas. Actually here, for instance, you see very nicely in front of that gene, you see the areas which are covered by those reads of this ATAC-seq experiment. And probably if you run this peak algorithm, this will be picked up as a peak. So I'll show you this that we have these peak calling algorithms here, and one of the most, the one which I presented for the cut-and-run, we haven't yet implemented here in the platform, but the standard one, which is a max algorithm, we have here. And basically what you do is, let me actually decrease this a little bit so you see it bigger. Maybe I will make it a bit bigger so you can see it more cleaner. So what you do, you actually take your BOM file, for instance, this one, and you can define also the background if you have, and then you create your peak, and you can specify here the parameters of the peak calling. So what I did, and then here are the peaks. So if you drag and drop here the peaks, you can see that those areas, for instance, this one is covered by, was found as a peak. So let me basically see it more clearly. So it's a little bit, I will close this just to show you a bit more on the screen. Yes, here we see the peaks. And let me just, so this is another peak. So you can zoom out to see where are those peaks in the genome more clearly, right? So you can go through the genome and see where are the peaks. All right, I think I will drag those peaks. Let me put maybe more peaks here. Also from here. All right. So I will take those peaks above, just to put it a bit above, so to see them better. So they're here. So you see these peaks, they are not only in front of the genes, sometimes they're inside the genes. So in introns and the enhancers, the open chromatins which contain enhancer can have those peaks. All right. So tell me whether which kind of peak calling algorithms you are using. There are a number of them which I showed. If you are using any, just type here in the chat. So I'm just interested. If you are always using MAX2, which I also like, or MAX, older version of MAX. Yeah, you see people are actually also using MAX, but some prefer other types of algorithms, and it's always interesting to compare what types of algorithms you are using and the results from those different algorithms, because actually the peaks are some sort of artificial things, because in fact the experimental facts are those reads. So where there was an observation that there was actually the transcription factor sitting, maybe very rare, where very little probability, but it was there. So the peaks is just the positions of the most possible sitting of those factors. So they are dynamic, they are coming, they are going away. So that's why different algorithms, they can give a different result. Anyway, so we have those peaks. And okay, I will now go a bit further to tell you what we are going to do with those peaks, right? So we are going to, as I said, we are going to figure out which transcription factors are going to bind to these ATAC-seq areas. So let me see whether my program has finished. Not yet. So all right. So actually, let me just go further with my slides a little bit. So it's actually showing just one slide here. So let's consider this transcription factor BPTF. So how do we know where these factors can be bound? So first you need is to collect, actually we did it for you. So we have a TRANSFAC database, and we went to different, we began shopping. So we went different papers or different databases, or just raw data of any ATAC-seq or any ChIP-seq experiments. And we collect data about known binding sites for this transcription factor. And we prefer those sites which actually really experimentally confirmed. And the better confirmation that it's binding site, it's not only Sometimes, especially in older time, in old good time, there were a lot of researchers confirming those binding sites with many different functional assays. So telling it's not only binding, but it's also contributing to the expression of a particular gene. Or maybe it's done with some in vitro experiment when there's a transcription factor and with a CELEX experiment, for instance, you do a lot of oligonucleotides across this factor and you select those oligos, and they also show to you which kind of sites can bind to this protein, although maybe not in vivo, but in vitro. Still, this kind of data are collected, and then we build a matrix. So this is a positional, so-called positional weight matrix. So we align those known sites, and we simply count how many times a particular nucleotide was observed in particular position. In case of BPTF, this is a very kind of simple, in a sense, a motif with a lot of A. So although there are some C's in between, but those two A's in the center, they are more conserved, but the other nucleotides around are less conserved. So then what happens next? We take this matrix and we apply it with certain formulas to scan through the any sequence of our interest. And we call this a new transcription factor binding site, which is not yet known, but potential binding when this score of binding is higher than a particular threshold. How do we know which threshold is there? It's a whole story. How we define those thresholds. Actually, they are defined automatically in our system, but we also have predefined thresholds to show you, to give you a most probable binding site, so less probable, but not to miss any potential binding sites. So you can have a higher threshold or lower thresholds. So that's how we predict binding sites for the transcription factors. All right. But now coming back to this tool. So let me basically first work a bit with these motifs and with the peaks. As you can see, we had those peaks which are for the wild type, and the peaks which were for the knockout. For instance, these ones 6, 7, 8, 9, they are for knockouts. So if we put them here, it's already there. So let me remove this track, or just actually not, just to show you a little bit better. So we have here the peaks for the wild type, and here we have the peaks for the knockout. And you see some of the peaks are the same. So which means the open chromatin was the same in the wild type and in knockout. Probably this chromatin is kind of always needed to for the cell to function, right? But some peaks are here in knockout, but not present in wild type. In other way around, maybe you can see some peaks which are present in wild type, for instance, these ones, but not present in knockout, which means these were open chromatin in a normal cell, in this acute myeloma cell, but when there is no factor of our interest, there is no peak. So which means probably this factor is actually binding there, right? So let's then combine all those peaks together from wild type and from knockout by joining this peak together. So actually, in our tool, I of course did this joining, but one nice thing here is when you click on the ready result, like this is joined peaks for wild type, you actually see which type of function was used and what kind of parameters. The only thing you need is just click here, and then this particular function of join track is opened. You can use it again. So basically, I put here all the peaks by collecting them from this folder and simply running this join track. And then I got this join track here, so which joins all the peaks from all the all 4 wild type experiments. And the same I did for basically all the peaks for the knockout experiment. So they are here, joint peaks for knockout. So there are also quite a number of them. All right, so And some of them, then what I wanted to do is actually, you see there are bigger peaks, smaller peaks, they depending on the experiment, and they overlapping. So actually, I want to find the defined areas and also of a definite size. So I want to kind of minimize those peaks to a particular size to analyze maybe centers of those peaks. And for that, I have this function called actually process track with sites, where you can say here my joint peaks, and then I actually want to shrink them to the summit, because each peak has a summit. Summit is the top of the where you have the maximum number of reads stacking to each other. And then you first shrink this peak to the summit, and then you extend it 300 nucleotides up and left and right. So basically, you create all the tracks of the size 600 around those peaks. So what we can see here is those peaks of the length of 600. So you see them here. So they are well defined. The same I did for the knockout peaks. And now what I want to do is actually to find the difference between these peaks, which are found in the wild type, and remove those which are found in the knockout. So these ones. So you see, I have those ones in the wild type, and I want to remove those which are in knockout. And for that, I have this intersection function. Here it is. So let me intersect tracks. So actually what I did now, I put here those first peaks, the second peaks, and now it intersects, and now I see the difference. So now if I put here these different tracks here, so I see only few tracks which are really different. So in fact, when you look at how many peaks do we have which are different, it's 22,000 peaks, although initially the wild type peaks, it's actually 474,000. So a lot of peaks, in fact, are common, but very few are really specific for the wild type compared to the knockout. All right, so now we have those peaks. So you see, you need a lot of pre-processing before you really go to find those motifs. Actually, what we are going to do now, we are going to go here to the ChIP-seq analysis. But in fact, what I did to run it faster, I choose not all 20,000 peaks. Of course, you can do it like that. And in fact, as you can see, I have here among my processes, I currently have my processor still running, which analyzing all these 22,000 peaks. It's a heavy task because you have, just to remind you, we have now in TRANSFAC, well, if we talk about vertebrate profile, so vertebrate matrices, we have around, I think around 8,000. Actually, we can have a look more exactly. If you look here in the databases in our system, so and look at the TRANSFAC, the latest release, you see here the profiles, meaning the collection of matrices for different organisms and for different situations. And if you look at this collection, so of the matrices, so we have a bit more than 8,000 positional weight matrices for vertebrates. So all of them can be applied to find motifs for transcription factors working in human. So you have a lot of things to do. So to analyze enrichment of all these 8,000 motifs. So it's a pretty big task. That's why what I want to do first is to find a smaller task for us. And what I actually did, I selected a very small subset of those 400,000 peaks. I selected 15,000 peaks. We can open them here, which have a maximum score. So those which have a maximum top score of the value, the most amount of reads are there. So we have this, well, actually it's around 20,000 peaks. which I choose to analyze, but these are wild-type peaks. I want to find those ones which are specific. So I intersect them with the knockout peaks. And here I have peaks actually which are different. And these are only 52 peaks which have maximum high value, and they are different from knockout. So these are peaks we want to analyze. And that's what we are going to do. But let me go now back to this section, and we go to ChIP-seq analysis. And now we go to the analyze ChIP-seq peaks, and we want to actually identify enriched TFBS in these ChIP-seq peaks using TRANSFAC, of course. And we click here, and what it shows to me is actually shows the whole pipeline. So in fact, this system is not just running individual programs. It can run the whole pipelines, and actually you can also edit those pipelines, adding new parameters, adding new functions to it, and so on. So that's a special story how to work with pipelines. But here we have a ready pipeline. So what it's going to do, it will actually take those peaks. So I will take these peaks from here, and I will tell, okay, this is a human. And then actually it will create a random track. So it will pick up random track from a whole genome, but from regulatory part of genome, it will take a random track, pick up random regions from promoters of many different genes. So actually what it's doing, randomly just pick regions of the same size from promoter areas of genes. And then it will run motif analysis in our peaks, and we'll run it in these random peaks, and then we'll compare those two results to each other, and then we'll see the results. So that's why actually what we want to do is we want to run a small redundant set of, I would better actually go to the full one. So we have here different types of profiles. For instance, there's a very small profile of non-redundant kind of profile of very small set of transcription factor binding site motifs just for the first look, but we may miss very interesting transcription factors. So that's why I would go for the full analysis. Actually, right, so just take it on the call here, and basically it's ready. So then let me run it. So it will take some time. So you see it's running this this site search in my tracks, and it's running then it will run the random tracks simultaneously, and then it will start comparing them. All right, so you see here, it will take maybe whatever 5-10 minutes until it's ready. So you have now times to ask your questions, and I see already some of your questions in Q&A section. I have one question. What do you mean by join or joint peaks as can be seen in the data panel? All right, again, so all right, so also there was a question to increase the size, the screen resolution. I hope it's better now. I already increased it. All right, but answering this first question, joining the peaks. All right, so that's again what we are doing. You see, we have the peaks from this individual experiment. So this first peak set, this second set of peaks, these are individual experiments. So right, in the case of that experiment, there was basically, if you look here, there were several while they run in parallel, several I don't remember actually, there were 4 independent experiments on 4 different cell, well, it's the same cell line, but they run it 4 times. So they run the wild type, and then they run 4 times knockout. So you have these replicas of the same experiment. So that's why I have here all these peak collection coming from each individual experiment. And then I simply join all the peaks together. So as we have seen here, so these are peaks coming from one experiment, and then coming from another experiment, for instance, from this one. And they are all from the wild type. And then I join them all together in one function. And then you see some of them overlap, many of them, because they're coming from the same type of experiment, so from wild type, but they might be different. So because individual actually cells and individual experiments may run a little bit different, right? So you have always variability, like in this case, for instance, in these experiments, this is the same peak, but it's a little bit shifted left in these other two experiments. That's why you see here in this collection, this bit of crowd. That's why you have to minimize them by joining those peaks together. Well, kind of, if you have these two peaks, instead you get one peak, which is joining them together. All right, I hope I explained it clearly now. Right, so I have another question. How to get the unique peaks in wild type? And yes, that's exactly what I was describing. So I run this process called intersect tracks. And then I put here the peaks, these joint peaks from the wild type experiment, right? So these ones. And I compared them with joint peaks from a knockout experiment. So, and this function is called, intersect tracks, and the output is actually you have two outputs. One is shows you intersection, so actually shows the common peaks, and you see here 145,000 peaks which are common between wild type and knockout, and you see also the difference. All right. Okay, so we see it's still running. So our analysis. So I will proceed now with the second example, and in the end, when it's finished, I will actually tell you whether we found the motif we are looking for or not. Okay, so the second example I want to present is the example of CUT&RUN. And in that case, so I guess you see now, again, my my presentation. This is another paper of another, but very similar experiment. Actually, it's published very recently. And here we have another disease. Well, they studied another disease. It's a B-cell lymphoma. Actually, it's a diffuse large B-cell lymphoma. And in this case, one factor which is very important for this lymphoma is FOXO1. And they did the same. They actually knocked out this FOXO1, but they did it very nicely. They knock out it conditionally. So basically, you can knock out it at once, and then you see what happens in very short time. And then what they actually did, they did ChIP-seq with nucleosome acetylation marks, but they also done this CUT&RUN with a dry trimethylation mark, which are, as you remember, is actually markers of the promoter areas. So rather than enhancers, right? So but they actually present also in enhancers, but they're more specific for the promoter areas. Okay, there's another question. I will answer them in a moment. And so we can use this data for further analysis. But what was also interesting, they also had an RNA-seq experiment. So actually done on the same set of cells. So in the same cells, they did CUT&RUN and RNA-seq. So which means they know which areas are open, and not only, well, which areas have this methylation marker, which is a marker of active enhancer, active promoter, and a bit of enhancer as well. But also they know which genes are upregulated. So you have two types of omics data, epigenomics data and transcriptomics data. And our tool allows to combine these two types of data in one run. And what we actually, and that's one thing. And the second thing which we are doing in our tool is not only just searching for motif enrichment, but we are searching for a combination of motifs. And that's, very specific type of analysis is done using genetic algorithm. It's a bit of machine learning, and some people call it artificial intelligence. So, but basically what it's trying to find, it looks at the promoters, because we are talking now about promoters, right? Promoter areas of upregulated genes, and it looks for the motifs which happens to be in clusters. So different motifs for different transcription factors, and then clusters. Why in clusters? Although the knockout was just for one factor like FOXO1, but as you can see, as you will see now, this FOXO1 is actually not working alone. It's working together with other factors, and when it's knocked out, then the whole complex of factors is evaporated, so it's not working any longer. And that's why we see or not see those complexes of factors in these areas of interest. So that's how we're going to work. And for that, we have another tool here. And I will go now to another folder where we analyze this data. We analyze this data. This is here is in this folder, right? So here what we have is two types of data, as I said. One is this already, they did the peaks. So we are not calling here peaks ourselves. We simply upload the ready peaks. So we have here 29,000 peaks of the length, all of them are lengths of 400. So the authors, they actually specify those peaks very sharply. So just 400 nucleotides in the center of the real areas, which were found by CUT&RUN, but that's okay. So that's the peaks we are going to work. And then we have transcriptomics data. So here we have a transcriptomics data where 20,000 genes were measured actually in the time zero. So before they did conditional knockout, and after 6 hours of the conditional knockout of the FOXO1 transcription factor. So and then so they did 2 experiments in time 0 and 2 experiments after 6 hours. So we have this data and we want to combine it. So of course there are many steps to do that, but we have here a very nice tool which is called actually Genome Enhancer, which can do it absolutely automatically for you. The only thing you need is actually to drag and drop here in this box your peaks and your RNA-seq data in form of these counts, or in the case of which I presented, it was FPKM values from RNA-seq experiment. And then, when you proceed, I will show how then it looks like. You specify here. You specify here 2 conditions at time 0, for instance, and at time 6 hours, whether you choose FPKM or counts, that's your choice. And then you have these two experiments in time 0, 2 experiments in time 6. And you have these peaks which were measured actually in time 0. So, and then you simply proceed. And then here you specify your disease, which you are studying. So it was this lymphoma, large B-cell diffuse. And then you select which conditions you want to compare. And we compare T0, because it was before knockout, to the T6 after knockout. And then we start analysis. It takes around 2-3 hours to finish because it's doing a lot of comparison. What it's doing first, it takes genes, but we'll see it right now in the report. So actually it generates a nice report at the end like this. So it reports you actually the most interesting finding in the first sentence. It identifies this gene, EZR, and we'll see it in a moment. It's actually very interesting as a master regulator. And it also finds actually FOXO1 as the most important transcription factor as in this case, together with two other, twist and rel. And it also tells that this master regulator can be important for treating as a drug target is treating lymphoma, especially B-cell lymphoma and diffuse large B-cell lymphoma. So first, what I want to show, it gives a lot of interesting insights, what it's actually doing. Just briefly, what it's doing, it's first it compares this T0 to T6 in terms of RNA-seq, and it finds which genes are upregulated and downregulated. Then it takes those peaks and starts searching whether those peaks are around those upregulated or downregulated genes. It's also mapping them on different types of ontologies, on geoterms. But this is something what is very specific, and that's about these clusters of sites. Then it finds, using this genetic algorithm, that this combination of motifs is significantly enriched in the peaks, which are, in a wild type, well, in the time 0 in this case, right? And they are different from the random peaks. So the time 0 peaks are enriched by this combination of motifs. You can see it here in more details, actually on such a picture. It will be generating. Well, while it's generating, let me answer your question. There is a question here in the chat. On which basis we will call a peak as an open or close? How to make a list of open and closed? All right, that's maybe a mix of the definitions. So when we are talking about ATAC-seq experiment, right? So actually the experiment itself, because it uses this transposase, which cuts the open chromatin, so which means DNA is open, naked, sort of. There's no nucleosomes, nothing covers it. Then once you see the peak, it's by definition is open. This is open chromatin, right? All peaks are showing open chromatin. Maybe I used like open and closed. No, there is a open chromatin and closed chromatin. The closed chromatin is covered by nucleosomes. So which means there will be no access by those peaks, right? By this transposase, and there will be no peaks there. So when you see the peaks, this is open chromatin. But then you need to compare. So you have open chromatin in one situation, like for instance, in time 0, and you have situation in time 6, and then you compare. So you have two types of open chromatin, and in the difference, you see very specific open chromatin, which basically, well, I think my system is very busy now because actually I run two runs. All right, it's now opening the report. Okay, so I see what's happening. It's actually finishing my run, which was started just before. That's why it's kind of got frozen, but we are now here in my report. So I will actually close this. I will open it again. Well, it's not opening. I think it's still busy. All right, so I will anyway go back to open this further. So as soon as this process is finished, I will show you those clusters. But here what we see is how those clusters are different from the random regions in genome. So on this histogram, what it shows how many clusters of these motifs you observed in the areas areas of trimethylation in front of upregulated genes in T0 time point. So which means these are very specific areas of active areas of promoters in front of upregulated genes, which are occupied by these factors, including FOXO1, as it is shows here. And which disappears, these areas disappears when FOXO1 is knocked out. All right. So which means this FOXO1 indeed is very important. And together this, and it works to activate those genes not alone, but together with these other factors, including CRL, so it's actually NF-kappa B binding transcription factor, SMAD4, and other transcription factors. So they form complexes to upregulate these genes. All right, so now we know this complex of factors. Let me go further and show you what else we can do once we find those factors. So basically what you see here, we see those upregulated genes here, which you found in the experiment. And then we see these factors. Actually, let me open this diagram a bit bigger. So here below you see these genes which are upregulated in the T0 and which goes down after knockout of FOXO1. And then these are the transcription factors, the blue ones, the transcription factors which are actually working on those genes, activating those genes. And this upstream from factors, the green and the red molecules are the regulators in signal transduction, which regulate activity of these factors. So what we have in our tool, we have this algorithm which goes upstream from these factors and it finds so-called master regulators. So among them, we see like interleukin-4, for instance, or BMP7, and you see this erzine, and we'll talk a little bit about it. And also this IgB is actually very important antigen in leukemia cells, which indeed is not only a known key master regulator of those cells, but also it's a good drug target, which is also shown here. So in our report, we show also research for the drug targets. And it shows that this IgB, it's also another name, it's a gene name for the IgB. This is, very specific antigen for those types of cells. It's a known drug target of a drug. And actually, we also see here this erzine, which also has a drug on the market, although not for leukemia. And it's also, the system specifies it as an important master regulator here. All right. So actually, it also goes further. It also gives you a table of known drugs clinically proved or, which are in the clinical trials. And here you see against these master regulators, there is already clinically proved drug is exactly for large B-cell diffuse lymphoma. So basically, we predict what is already known just from RNA-seq experiment of this knockout RNA-seq experiment plus CUT&RUN experiment, nothing else, no additional knowledge is what's put in the system, but it automatically finds the correct drug targets and even predict the correct drugs which can be used here to inhibit activity of those factors. So instead of basically knockouting of FOXO1, you can inhibit the activator of FOXO1 together with cofactors, which will repress those genes, the oncogenes, and basically sends these cells to apoptosis. So these drugs actually are already used. And as for these very nice prediction of erzine, and there is a known drug auronafin. Actually, it's a completely drug which is not used yet in clinics for the leukemia and also for cancer. It's actually it's immune suppression drug, but here the system predicts it's like a a potential repurposing drug for this case as well. All right, so that's the whole story, how you can just start from RNA-seq and cut and run experiment and build the whole story about which master regulators are important, which drug can be used, and which, of course, which transcription factors are important there. And one little note here, of course, I looked in this paper where authors of this data, they analyzed the data by themselves, but they didn't use Genome Enhancer. And they actually was not able to find the FOXO1 motif. So they reported in the paper that unfortunately they cannot see the FOXO1 motif over there. So they used some other tools to find the motifs, not TRANSFAC. That's first thing, but most important, they used just for a single motif. But here, appear to be important a combination of motifs, which together supports of FOXO1, and then you see it in the results. All right, so we'll now return to results of the first experiment, but first let me answer your question. In one slide, it was mentioned CUT&RUN seq is cost effective than ChIP-seq. Could you please emphasize this point a bit? Okay, so I'm not expert in the experimental part, but that's just what I read, and actually it makes sense. So, as I said, the CUT&RUN, actually, because it doesn't have this step of cross-linking to DNA, which produce a lot of artifacts. The cross-link itself, it's known in, in ChIP-seq experiments, the cross-linking produce a lot of artifacts. And here there's no cross-linking. That's why there's no very little noise. And since there is not very much noise, you don't need to do very deep sequencing. So your sequence depths could be much less than in case of ChIP-seq. And then when you do less sequencing, of course, it's cheaper. So just proportionally, you can run on the same sequencer run much more, experiments, and altogether it will be much cheaper. That's only one part of the that cost. But as I said, I'm not an expert in the experimental part. You better ask those who really do it experimentally. All right. So and now we are going to go back to our platform. And by the way, you see this, I'm switching back and forth from platform to the Genome Enhancer, because the platform is indeed a platform. So it has a lot of function, a lot of programs inside. It's more than 200, I think, programs and a lot of pipelines. But we have these additional tools like Genome Enhancer, like Match Suite. Here, we have Match Suite, which just one pipeline on top of that platform with this special interface, an easy interface where you can simply drag and drop and simply run like with a few clicks. But of course, you have to be careful, because you sometimes you would like to choose a different cutoffs, different thresholds in your selection of genes. In our pipeline, like in Genome Enhancer, there's all predefined. You cannot change it. So if you want to run it with your own parameters, then you actually go back to platform, and then you have to run all individual tools like I did in the very beginning, integrate the peak together, remove one peaks from another, and search motifs in them. So that's you have to do step by steps, but then you are all controlling all your parameters here. All right, so now we go back to this AML example, which is here. And as you remember, we were running this different. So we were running site search, enrichment site search on these 52 peaks, which have a top value of the enrichment by reads. So it's the highest peaks in genome. And then here we have these different, and here we have the site summary. All right, so what you see here, here we are not yet doing any combination of motifs, just individual motifs by themselves. And you remember we run all 8,000 motifs, and here you have a list of 2,700 motifs which got enriched in these 52 peaks. And enrichment is shown by two parameters. First is yes-no ratio, which is showing how more frequent is that peak, that motif in these peaks compared to the random peaks in the genome. So for instance, Krebs motifs are 38 times higher in frequency in my peaks compared to the random peaks, and the p-value is significant. But for us, what is important is actually the motif for the BPTF. And if we look now for that motif, and I have opened here this entry of BPTF factor motif, which is here. This you've seen, you remember from my slides, this was the motif, but actually motif has a different name. It's called FAC1. So the factors and many genes have different synonyms, but this is exactly BPTF. And this motif was from Selleck's experiment. So that was one entry which we put quite from very old, it's a 1999 paper, but we still have it, and it's a true motif. So let's find this motif in my results. So let me see, and as you know, I am now a bit nervous because actually I didn't run it before. No, actually we don't find this motif. So which means in the top peaks, the enriched motif is not there. The FAC1 is not among enriched motifs. So which means the highest peaks, which actually found by this ATAC-seq experiment, are occupied not by FAC1, not by this factor. Just to tell you that when I ran for the whole 22,000 peaks, I found a significant enrichment for that factor, but it was not among the topest peaks. So which means this the story here is much more complex. But which peaks are most enriched? Which motifs are most enriched here? For instance, if we could look at this CRAP or maybe an F-kappa B, or take them all these top ones together, then we can actually look at them where they are located. All right, so well, in this case, they are together. So you see, these are the, so what you see here. So this is one particular region, one peak, right? In this gene, so inside certain gene, in a particular area in genome. And in this peak, we found sites for CREP1. This is HLF sites, the CREP ones, I guess. These the gray ones, I guess. Yes, the CREP BP1s are these ones, the gray ones. And we found here in this peak and this peak and in this peak. But there are very many peaks where we didn't find. So this motif is enriched, but it's pretty rare among the peaks. But if we actually sort by p-value and let's select those motifs which are more frequent. For instance, this motif is more frequent, and you see it's among our peaks, it's like approximately two motifs per peak, But in the random matrix in genome, we found much fewer of these motifs. So if we visualize that, so you see that there are peaks which are actually quite full of those motifs inside a short area. So that's why these areas are attracted by this transcription factor, and it occupies these areas and remove the chromatin, and that area becomes open. Whether it's contribute to the regulation of the gene, we don't know, because here we just take pure peaks and we analyze motifs. That's it. So then the conclusion we can see here whether we found peaks, we found motifs for particular factors or not. The conclusion about the regulation of genes and master regulators and all what I showed for the second example can be only done when you combine it with expression data. All right. Okay, so with that, I would like to finish for today. It was quite a lot to show. I hope you enjoyed and you understand how the system works. We still have time. I mean, as the session is open for a longer time. So if you have some questions, just write it here in chat or in Q&A, and I will try to answer them. And by the way, if you are watching this in YouTube, so you please put likes to this if you like it. And then because in YouTube we need those likes that many people see it, and I think they could find this useful. All right. So I'm returning back to the start of the page. As you can see, again, this system can analyze not only ChIP-seq or ATAC-seq or CUT&RUN, but it analyzes many other omics data, as we've seen today, RNA-seq, but also, for instance, genomic data. So if you have SNPs or other experiments, so you can use it here to analyze whether these SNPs changing transcription factor binding sites. You can analyze even the microRNA data, so to understand how microRNAs are regulated, because microRNA genes are actually also regulated by transcription factors. They have their own promoters, and that's very specific type of analysis you can do here with a platform. All right, so I think if you have still questions, all right, so I have a question here, the chat. All right, so that's if there are any specific question in the future, where can we post them? Please share the email. Thank you. All right, so yes, my email is this. Well, but actually, if you would like to send it really not only me, but actually let me give you my email. It's Alexander Kell at geneXplain.com. You can send it by email. Also, we have actually here the website where all our future seminars are presented. It's called geneXplain Academy. And here, yeah, you see here this geneXplain Academy. And here you also see which further next topics will be addressed in our next seminars. You already can book your seats. And here in the past session, you see what other past sessions were there. But also you can actually ask your question. All right, here, yeah, in here in the top of that and that field, right somewhere. Yes, here you can put your question. Please do that. So then not only me, but our team gets the question and you will get the clear response. Okay, I have another question. I have not been using my account for a while. Is it possible it has been some way disabled? All right, so it depends how old is your account. Well, actually, what you have to do is you simply go to our registration page here, register here, all right. And if you have if you have not registered yet, you put your license key and you register it again. So you have so if you are a customer, if you have your license. If not, you simply forgot your password, you click here, forgot my password, and it will be mailed to you. All right. Yeah, exactly. You see also in the comments this link to go to the user account, exactly this page. So in here, if you have a license key for the paid subscription, or you want to open the free platform account, it will be open for you automatically immediately. So you get this standard license key for the free platform, and you put your information, and you get your account again. So if you lost it, so then if you remember your email which you used to create your account, you simply can ask for forget password, or simply create a new account using a different email address. Thank you. I think we can finish for today if there is no more questions. And thank you very much for attendance and for your interest. Try platform. Yeah, you can do many things with a free platform account. We have a special seminar. We had a special seminar in which you can find here in the recording of that seminar about using the free platform. You know, you see, explore your free platform. We had this seminar some time ago. So you can do many things using free platform for for actually analyzing a few number of peaks. This platform has a limited space, so you will need get more space if you need more. Yeah, but if you have a license for TRANSFAC, just open your account and go ahead. All right. Okay, thank you very much, and see you next time. Goodbye.